Hi, I'm Beth Beluccio. I'm the Global Clinical Lead for Rare Neurological Diseases at Pfizer, and I'm really excited today to be able to talk to you about Pfizer's investigational gene therapy for Duchenne muscular dystrophy. We started investigating this gene therapy about 2 years ago in our phase 1B clinical trial, and that trial is ongoing. We're still enrolling patients and treating them. And last year at the Parent Project Muscular Dystrophy Connect conference, Michael Binks presented some preliminary efficacy and safety data from the study. We're going to present some new data on this study at a scientific conference later this year, so I'm not going to focus on that in my presentation today. Instead, what I want to focus on is our phase 3 clinical trial. This clinical trial was recently posted on clinicaltrials.gov, and so I wanted to have a chance to review with you a few of the details of the study. The study is designed as a global, multi-center, randomized, placebo-controlled, double-blind study. We have two main objectives for the study. One is to determine whether or not this investigational gene therapy is safe and effective for Duchenne patients. It's really important that we test this because until we run a big phase 3 study like this, we don't know for sure if it's safe and effective. The other main goal we have for the study is to very quickly and efficiently gather this data so that if the study does show it's safe and effective, we can take that data to regulators and hopefully allow them to approve the drug, which we think is the best way to make this available for as many patients as possible. We've designed our study with a placebo control arm, and we've done that for two reasons. One is it's the best way to have clear conclusions from the study about whether the drug is safe or effective. And the other is it aligns with expectations of the regulatory authorities. But to try and minimize the number of patients who have to be exposed to placebo, we've designed our trial with a 2-to-1 randomization ratio. That can be most easily explained with the diagram at the bottom. So as you can see, we expect to enroll approximately 99 patients, and 2/3 of them, or about 66 patients, will be assigned to cohort 1, and they'll receive gene therapy at the beginning of year 1. Approximately 1/3 of the patients, or 33, will be assigned to cohort 2, and they'll receive placebo at the beginning of year 1. And then about a year later, at the beginning of year 2, they'll be eligible for the gene therapy treatment. The patients who are randomized to cohort 1 will receive a placebo injection at the beginning of year 2. And this is really so that all the patients, whether they're in cohort 1 or cohort 2, get the same number of treatments at the same timing in the study so that no one can tell which cohort they've been assigned to. And that's part of keeping the study blind, which is a very important part of making sure that we can interpret the results correctly. Patients who are randomized to cohort 2 and receive the placebo at the beginning of year 1 will have to be tested for a few eligibility criteria prior to receiving their gene therapy at the beginning of year 2. And this is primarily to make sure that the gene therapy would still be safe for them. They will not have to meet all the inclusion and exclusion criteria for the study at the beginning of year 2. All of the patients who get randomized into the study will be followed for 5 years after receiving their gene therapy. So patients who are assigned to cohort 2 will be in the study for a total of 6 years. So one of the primary objectives of this study, as I mentioned, was to determine whether this investigational gene therapy is effective in Duchenne. And to do that, we've picked a primary endpoint as our main test. The primary endpoint we chose was the North Star Ambulatory Assessment. And what we'll be doing is looking at the change in that NSAA total score from baseline, right before patients are treated, until the end of their first year, or at week 52. Then we'll be comparing that change from baseline to the end of year 1 between our active gene therapy treated patients and the placebo-treated patients. We, of course, also will be determining the safety of the drug over that first year. We have several inclusion and exclusion criteria that will be applied to make sure that patients are eligible for this study. This lists the key inclusion criteria, and that means that patients have to have these criteria to be able to enroll in the study. The The first is a diagnosis of Duchenne as determined by genetic testing. If patients have had genetic testing before they come to the study, like through their own doctor, they can bring those tests and those will be used to evaluate the eligibility. Patients also have to be ambulatory, and we'll use certain tests in the protocol to define if they're ambulatory so that everybody has to meet the same definition. We have an age criteria that patients need to be between the ages of 4 and 7 years old, all the way up until their 8th birthday. And we've had a lot of questions around this age range and why we can't use a broader age range for the study. So I wanted to spend a little bit of time explaining our rationale for that. As I mentioned before, one of our key goals for the study is to determine as quickly and as efficiently as possible whether our drug is safe and effective. One of the best ways to do that is to make sure that the group that we're studying has very little variability among the boys in the study. When we look at natural history data and got expert opinions from leaders in the field, what we determined was that between the ages of 4 and 7, the disease progression is fairly similar among boys. There's not a lot of variability. Whereas once you include patients who are 8 years old and older, the disease progression begins to widen and vary among boys. And then it becomes very difficult if you're comparing one group of treated boys to a group in placebo when there's variability, because there's just a lot of overlap. So by using a group of patients between the ages of 4 and 7 where there isn't very much variability, you can more easily distinguish whether the gene therapy is different from the placebo and whether it's effective. We also are requiring that boys be on a stable dose of daily glucocorticoids for at least 3 months before coming to screening for the study. If they're on prednisone or prednisolone, the minimum dose that they need to be on is 0.5 milligrams per kilogram per day. And if they're on deflazacort, it needs to be at least 0.75 milligrams per kilogram per day. The reason for this requirement is, again, there's a lot of variability in the dose of steroids that patients take and the pattern that they take them. So again, if there's a lot of variability among the boys, it gets very hard to distinguish whether our gene therapy is effective compared to placebo. So we want to keep all the boys on a very similar regimen so that it's easier to compare our gene therapy. We also have several exclusion criteria, and these are criteria that a boy cannot have if they want to enter the study. The most important of these is a positive test for neutralizing antibodies to AAV9. If boys already have some antibodies to the AAV vector before getting gene therapy, the immune response is huge, and that can really put a patient at risk. So we don't want patients to have those neutralizing antibodies before being treated. There are a lot of different tests available for neutralizing antibodies, some that you can just get through your doctor, but the way these different tests determine whether someone's positive or negative differs. And so the only test that will be used to determine whether someone's eligible for our study is the test that's being conducted by Pfizer. We also have exclusion criteria that patients cannot have been on a therapy designed to increase dystrophin expression for the last 6 months before they come to screening. And these therapies include Translarna, Exondys 51, and Vyondys 53. They also cannot have received any type of gene therapy in the past. And then patients need to not have any injury that could interfere with their functional testing, such as the North Star Ambulatory Assessment, and they can't have any laboratory abnormalities that might indicate they have an underlying disease that would put them at risk for receiving gene therapy. So we're very excited that this trial is about to start. We have posted it on clinicaltrials.gov, and the NCT number listed here is one way that you can search for the study. You can also use keywords like Duchenne muscular dystrophy, gene therapy, and Pfizer as a way to find the study. When you get to the site and you see the study, there'll be a lot of details, such as the ones that I've been explaining. At this time, you won't see any of our sites listed, and that's for a couple of reasons. What we've done is we've explored a lot of sites, both in the US and outside the US, that we think are going to be excellent sites for our study. But there are a number of processes that we have to go through for each site before they can actually open and be ready to enroll subjects. Sometimes some of those processes are things we don't have a lot of control over. And these days with the coronavirus, we have even less control than usual. So sometimes things happen where we've identified a site that we think is going to be really great, but they're never really able to open. So we want to make sure that we don't really announce our sites until they're really ready to enroll patients. So as they become open, we'll be posting them on the clinicaltrials.gov site. The US sites are likely to open first. Over the first several months, we'll be adding more and more sites. The sites that are outside of the US will lag behind by a few months, and this is because there's extra regulatory processes that are required outside of the US, not only to open a study, but there's even additional things that are required for gene therapy trials, and this just takes a lot of time. Again, not really under our control, but as we're able to open sites, we'll post them on the clinicaltrials.gov website. So lastly, I just wanted to express our gratitude to the multiple advocacy groups that have been supporting us throughout the years, as well as patients and their families that we worked with very closely to try and understand the needs of the Duchenne population, to help us design our trials, and to help us understand how to support families both as they're deciding among different studies and as they enter a clinical trial. And finally, we really wanted to share our appreciation with patients and their families who do decide that they're willing to make the sacrifice and give their time and their energy to participate in our clinical trials. And with that, I'll thank you.